Section 8.4 is over transcription. Key concept is that transcription converts a gene, remember, which is a segment of a chromosome, into a single-stranded RNA molecule. We've talked a little bit about DNA. RNA is another nucleic acid, and its job varies depending on the type of RNA molecule it is. We are going to talk a little bit more about messenger RNA in this section. RNA carries DNA's instructions. The central dogma or theory states that information flows in one direction from DNA to RNA to proteins. Remember, proteins are important because they make up everything about you. Our DNA is located inside the nucleus where it can stay safe and undamaged. The RNA will make a copy of its DNA, carry it outside of the nucleus, find a protein, and make a protein. Seems kind of silly, but that's what happens. There are three processes in this. The first is replication, which we talked about in section 8.3. The DNA will replicate itself during cell division. So the DNA will make a copy of itself, the cell splits, and then the DNA is the same in every resulting cell. Transcription is the next process. We go from DNA to RNA during transcription. So you're transcribing the information on the DNA strand. And finally, because we're going from RNA and DNA into something completely different, a protein, we have to translate it, just like you're going into a different language. So replication, transcription, and translation make up the three processes of this central dogma. The RNA is a link between DNA and proteins. It differs from DNA in three major ways. The first, RNA has a ribose sugar. So DNA was made of a deoxyribonucleic acid, and RNA is a ribonucleic acid. RNA has uracil instead of thymine, and it's single-stranded, whereas DNA is double-stranded. So one of our nucleotides is going to be different. We have a uracil nucleotide instead of a thymine. Transcription will make three types of RNA. There's many more, but these are the only three that are important for you guys right now. Transcription will copy the DNA to make a strand of RNA. The three types of RNA that we talk about in biology are messenger RNA or mRNA, and its job is to carry information from DNA inside the nucleus out to a ribosome that's floating around in the cytoplasm. So you remember that messenger RNA carries the message from the DNA. Ribosomal RNA or rRNA combines with proteins to form ribosomes. So a ribosome makes proteins. Remember, we talked about that a long time ago. And then finally, transfer RNA, this fun little T-shaped molecule, tRNA, transfers the amino acids that are floating around in the cytoplasm, carries them to a ribosome, and helps match them into the coded mRNA message. Transcription is catalyzed by RNA polymerase. Remember, it ends in ASE, so it's an enzyme. Polymer means it is a complex chain made of monomers, and it happens to be RNA. This polymerase and other proteins form a transcription complex. This complex recognizes the start of a gene and unwinds a segment of it. So it's kind of like me telling you to start reading in your textbook on page 476 instead of just saying, hey, the information's somewhere in your book. So that start site, there's a specific start sequence that polymerase is looking for, and it tells it, hey, start reading here. And once you start reading, it's going to run across a start codon that tells it to start coding. And that's going to be important in this next section. Nucleotides will pair with one strand of the DNA, so it's very similar to replication. But the difference is, instead of replication going from DNA to DNA, transcription is going from DNA to RNA. That RNA polymerase will bond the nucleotides together. The DNA helix winds up again as the gene is transcribed, and nothing changes. We're not changing the DNA in any way. We're not doing anything significant. We're just opening it up, making a copy of it, and then closing it back up. The RNA strand detaches from the DNA once the gene is transcribed. So just like we said that there was a start codon or a starting sequence, there's going to be an ending sequence. It's not just going to keep reading forever. It's going to read from this point to this point. And that segment of RNA right there that you can see will go ahead and find a ribosome and make a protein. Ribosomal RNA forms parts of the ribosomes where proteins are made. So a ribosome is made of RNA. It uses mRNA and tRNA to make those proteins that make up everything about you. That tRNA brings the amino acids from the cytoplasm to a ribosome so they can be paired up. The transcription process is very similar to replication. They both involve complex enzymes, those polymerases, and they both involve complementary base pairing. The different end results are that replication will end up with two identical copies of the same strand of DNA. Transcription copies a gene. Replication makes one copy, and transcription can make many copies. So that one strand of mRNA is not limited to one-time use. It can be reused over and over and over by enzymes. You can see here there's one gene segment and the growing RNA strands that it's producing. So it can be producing multiple off of the same strand at any given time.